introducing a new subject. And let's see. Then I will have to give some introduction and hope I wouldn't run out of time as most of us here. In order to condense our few meter long DNA into the fraction of micron nucleus, we really it has to be compacted into very compact structure as the chromatin is. And if you will see here in electron microscope to the right, the condensed structure of the chromatin, and the cartoon shows it well. And then you will see on the right-hand side the relaxed form. The relaxed form is the acetylated form. Now, these forms are maintained partly and mostly by the form of acetylation. And if we will concentrate of the small unit, it composed of 4 pairs of histones that are wrapped with DNA. And the tails of them are rich with lysine, which has a positive charge, and by that the lysine, the histone, and the DNA are compacted to each other and attracted. When the lysine are acetylated, then the whole structure becomes more relaxed, and you can see that the chemical attraction is lost, and it allows many functions that needs that structure, access to repair enzyme and transcription enzyme, etc. It was shown that in cancer, tumor suppressor genes are being suppressed by compact structure, and their area, it has a lot of acetylated histone. So we have here a sort of pleiotropic mechanism. Drugs, if we will use histone deacetylase inhibitors, then we can take the, let me go backwards, we can take the condensed form that is present in cancer cells and shift it back to the loose form, the relaxed form, and thereby allow transcription of tumor suppressor genes. For that purpose, histone deacetylase inhibitors were developed, and we have developed a couple of them, which I will describe to you. Just to show you the structure in a more vivid terms, I brought this cartoon. And you can see that the end of the histones right here are being acetylated, methylated, ubiquitinated, phosphorylated, and there are a few other modifications that affect the structure and the function and the transcription of the chromatin. So our prodrugs are the first generation. I will not describe, there is a history, but I'll go right to the second generation, which is butyryl oxymethyl diethyl phosphate, which undergoes metabolic degradation in the cells to release the butyric acid, and the butyric acid by itself is a very weak histone deacetylase inhibitor, but we deliver it into the cells, and by that making it more potent. The second one that I will describe here is the derivative of histone deacetylase inhibitor, valproic acid, and acyclovir. So we have here a bifunctional prodrug that in the cells will release the acyclovir and the valproic acid, which is also known to be histone deacetylase inhibitor, and quite few clinical studies are conducted currently with valproic acid in cancer as a single agent and in combination with other chemotherapeutic agents. The their target are the histone deacetylases. Now, the histone deacetylases is actually 5 families of enzymes, which I don't show you the one that are NAD dependent, that all the SEERTs ones. And as you can see, they have a common structure, and they vary by location, activities, and a function. And the one that I will concentrate on are HDAC1 and HDAC2. It was shown by quite few reports that class 1 HDAC are elevated in cancer cells, and in a way they can use as a diagnostic tool to detect cancer cells, as well as prognostic tool, because the more it also the level of the HDAC of class 1 correlate with the aggressiveness of the cancer. So just how do our compound inhibit the enzyme? We composed a model based on the bacterial crystallized HDAC enzyme, and the compound can bind one moiety or two of valproic acid or butyric acid to the active sites that were the zinc binds, right in the middle by, as you can see, by the yellow. Here I demonstrate to you that in cancer cells there is inherent high level of HDAC activity, and it is demonstrated in the glioblastoma U251 and 41 cells, and in the cell line of cardiomyoblast, which is an immortalized cell. It is in between level, and in astrocytes primary culture that we isolate, it has a low intrinsic level of activity. We measured the effect of the prodrug, both of them, AN7 and AN446, on the activity using a fluorescent probe. And as you can see, the solid line are the effect of the prodrug on cancer cells, and the dashed line are their effect on the astrocytes. And the prodrug are selective in their inhibitory activity of the cancer cells versus the normal cells, and that correlates with their effect on their viability. The prodrug affect the viability of the cells significantly, to a greater extent that they affect the astrocytes, as you can see by the dashed line. So if we saw that we affect the activity of HDAC selectively in cancer cells versus normal cells, we looked at the expression. And what we saw was that initially the expression of HDAC1 and HDAC2 is higher in the cancer cells, as you can see in the left-hand side in U251, and lower in astrocytes. And the prodrug marked by, I don't know if I can use that, are really further decreased by AN7 and AN446, which do not decrease the level of this enzyme in the normal cells. So here we see that not only the prodrug affect the activity of the enzyme, they also affect the expression of the enzyme. And you can see it by the lines that demonstrate that. When we use the proteome to try and elucidate how the prodrug affect the level of the enzyme, we use the proteasome inhibitor, MG132, and we saw that the proteasome really abrogated the reduction in the level of expression of this enzyme in the cancer cells. And you can see that the level of RNA, which we looked by real-time PCR, was unaffected, while the level of the protein, that you can see here and here, while the level of the protein was significantly reduced by the prodrug and abrogated by inhibition of the proteasome. So one of the mechanisms by which the cells affect, by which the product affect cancer cells is enhancing their degradation, reducing their half-life, but by degradation in the proteasome. In glioblastoma xenograft, we wanted to get the proof in vivo. We looked at this is a xenograft of U251 in nude mice, and when we looked at the survival, we can see that the cell, basically, the untreated cells, they died, the blue line, they died from the tumor. The red line showed that doxorubicin, the mice died because of the toxicity of doxorubicin, because as you will see, tumor burden is reduced. The prodrug are shown that they do not affect the survival, they increase the survival of the mice, as you can see right above, no mortality, and furthermore, they protect the mice when we add them to doxorubicin treatment. They protect the mice from doxorubicin toxicity. And when we looked at the ratio of the heart weight to the body weight, which if it increases, it indicates cardiotoxicity, we see that the doxorubicin increased the ratio, whereas the combination of our prodrug with doxorubicin abrogate this cardiotoxicity. And we, in the paper that you see here, +1, we describe it in detail in a different model of mice in 41. The tumor burden is decreased by all treatments, as you can see here, and but you have to remember that doxorubicin mice lived shorter time, so the tumor didn't develop. We compared our compound to SAHA, and we saw actually that SAHA was quite toxic to the animals, and although it reduced the tumor burden, it did not protect the survivor of the mice. And our compound acted to potentiate the doxorubicin activity. So we can see here that while we protected the heart from the cardiotoxicity of doxorubicin, we enhanced its anti-cancer activity. So we see here selectivity in vivo as well. We looked at the activity of HDAC in the tumor and in other tissues, and I have to tell you that we loaded different amount of protein because the HDAC are expressed the highest in tumor. We loaded twice as much here than in brain, and twice as much in heart than in brain. So in all tissues, the treatment with the prodrug enhanced the acetylation of histones, as you can see here. The prodrug increased acetylation, so that means that they inhibited HDAC activity. Inhibition of HDAC activity increased hyperacetylation of histones. And when we looked at the expression, we saw that the compound specifically reduced the expression of HDAC1 and HDAC2 in the tumors and did not affect them in the heart. And furthermore, if you will see the peculiar phenomenon, doxorubicin enhances HDAC1 and HDAC2 activity, but it's an indication of cardiomyopathy, whereas our compound decreased this abnormal increase. So you can see here that the expression of of the HDAC1 and HDAC2 was reduced in the tumor and was unchanged by the compound in the heart, while in the heart, the compound reduced the abnormal level, which was induced by doxorubicin due to cardiomyopathy, and again, supporting their protective effect. So we looked at acetylation, at HDAC inhibition activity, and the effect on the activity and the expression of the HDAC1 and HDAC2 enzymes that affected specifically by the prodrug in cancer cells and in tumors and not in normal tissue. And the second aspect and mechanism by which the prodrug worked that we investigated was DNA damage and repair, DNA damage response. And as you can see from our result, when we looked at the tumor, the DNA double-strand breaks are marked by phospho-H2AX, which is increased by both the prodrug and doxorubicin, and the combination of them has greater effect on the damage. When we looked at the markers that participate in the repair complex, RAD51 and COO70 and RE11, we see that the compound, the prodrug, reduced them as well, and doxorubicin only affected the level of RE. So the compound increased the damage in the tumor by also reducing the factors that are part of the repair mechanism of double-strand break. So when we look at the heart, we see quite the opposite picture. While the compound, the prodrug, did not induce any damage as marked by phospho-H2AX, doxorubicin increased the damage and the compound attenuated this increase, again supporting the protective activity. Furthermore, while doxorubicin reduced the repair factors, and our compound either by themselves affected them somewhat, but increased them significantly when there was a genotoxic stress of the doxorubicin. So the link between, now I want to link the two mechanisms that I described, the acetylation, the inhibition of histone deacetylase, and DNA repair. Because if you already saw that we get acetylation, increase of histone acetylation in all tissues. So where is the selectivity? How do we affect the different tissue in a different way? And to that end, we looked at on selective acetylation. We looked at acetylation of lysine 56 in histone 3, and we used a kit, a fluorescent kit, to do that. And as you can see, the activity was inhibited. The level of K56 was inhibited in the tumor, the blue columns, and significantly. It was unchanged in the brain, and in the heart, doxorubicin increased it, while our prodrugs decreased the abnormal increase of doxorubicin, but increased it relative to naive mice. So what is the mechanism here? The lysine 56 is actually a gatekeeper. It sits right in, you'll see it better in the next cartoon. It sits, you can see, because it sits right behind the DNA there, and and it controls the accessibility of DNA repair factors. And by acetylating it, we allow the loose and relaxed formation that can increase repair. Therefore, it was decreased in the tumor and was enhanced in the heart, in the heart to allow repair in the heart and to avoid repair in the tumor. So we have here a selective mechanism. If I have a little bit of time, I'll talk about the selectivity that we see in T-cell cutaneous lymphoma, and it is a disease that when it caught up early, it can be treated, but when it's caught in this state, it's very hard to treat it, and the patients are treated with chemotherapy and radiation, and they large percentage of them relapse. Here we demonstrate with AN7 and then also with the trick was done to all of us, huh? That the product affects selectively the cutaneous lymphoma cells and affected to much lower extent than the lymphocytes when they're activated by PHA or not. Whereas SAHA, which is the drug that was approved to treat cutaneous lymphoma, exhibit the opposite selectivity. It affects mostly lymphocytes and to a lesser extent the cancer cells. So we hope to get with this model to the in vivo and to the clinic eventually. I probably will not have time to cover that. I just want to say that AN446, which has acyclovir in its structure, affects selectively EBV-associated malignancies, and EBV-associated malignancies are a major problem. The nasopharyngeal carcinoma, 95% of them are EBV positive. Gastric carcinoma, at least 10% of them. And our drug affect them, select the cells that harbor EBV, they affect selectively. So, to summarize the data that I presented, the HDAC inhibitors prodrug that were described target high HDAC activity and overexpression of this enzyme in cancer cells and in tumor. These effects are in accordance with their ability to induce cancer cell mortality and shrinkage of tumor. The selectivity of their effect can be rationalized by the inherent difference in histone modification pattern in normal and in malignant cells, such as inherent high level of HDAC class 1 activity and high level of their expression. And we see specific selectivity by the fact, if I didn't emphasize it enough, that in tumor, while we increase overall total acetylation of H3, we specifically decrease the acetylation of lysine H3K56 that plays a major role in DNA repair. AN7 and AN446 play selective and sometimes opposing biological roles in cancer versus normal cells, and I demonstrated it by the acetylation and the DNA repair. And they are selective. They are more selective than SAHA, which is an HDAC inhibitor that currently used to treat cutaneous T-cell lymphoma. And these properties altogether make them an attractive candidate for cancer treatment. And I want to conclude by thanking you and the people in my lab, Natalie Tarasenko, Gabriela Rosic, Nina Boyer, Bill Berkovich, Karmit Alessi, Alexenberg, and my collaborator, my longtime collaborator, Professor Abraham Nudelman, my colleague Kessler Isaacson, the physician that I work with, Emilia Hodak and Lilach Moyal on cutaneous T-cell lymphoma, and my collaborator in Australia, which I collaborated on interaction with Dr. Rubisi, and my support. Thank you.
What is the effect of these inhibitors on other